Namaste children. I hope everyone is fine and safe at home. Myself Madhvi, I'll be taking biology to you all. Today let us study about cells. So what is cell? The cell is a Latin word meaning little room. When I say little room, it should be somewhat small compartments. So this is a slide where you can see some cells. Okay, the cell is a Latin word meaning little room. Robert Hooke, an English naturalist, discovered the cell in the early 1665. How did he discover it? He took a slice of cork. Cork is nothing but a hard substance which is present in the bark of the tree. So when he took a piece of it and started observing under the self-designed microscope, in those days the microscope was so much familiar to the people. So it was not so much known to the people. So he designed himself a microscope and started observing under it. When he took a slice of cork and observed, he could see the structures like this. Okay, when he saw the structures like this, it resembled the honeycomb structures. What is this honeycomb? You might have seen where the honeybees keep its honey inside. So inside a little compartment, right? So it resembled the cells, right? So the little rooms or little compartments. The cell is little room. So next, what is microscope? A microscope is an instrument used to observe the micro objects that are invisible to our naked eyes. That means they are completely not visible to our eyes. So, such objects or organisms or specimens are observed under this microscope. The micro, when I say there are two things, that is micro and macro. Micro are very minute objects or organisms which are not visible to our eyes. Say, for example, bacteria. Can you see the bacteria? No. The virus, coronavirus, can you see coronavirus? So, such are the examples, very microorganisms. Micro, when I say they are tiny objects or tiny organisms which are not visible to our eyes. Such objects are the observed under microscope. Macro, when I say bigger objects, say for example plants, animals, you can see the animals, bigger animals, right, which are larger in size, can be seen through our eyes, whereas minute objects are not seen. So such objects are observed under this instrument called as microscope. Okay, so let us move to the next slide. Microscope. Microscope was invented by Janssen Hans Lippershey. Janssen Hans, our father and son team together invented microscope. And this microscope has a unit to express the size of the cell. That is, 1 micron is equal to 10 power minus 6 meter. That is, 1 micron is 1 millionth of meter. So this is expressed in 1 micron. And generally, there are two types of microscope, that is compound microscope and electron microscope. Here is it. This is compound microscope and this is electron microscope. Compound microscope magnifies the object about 2000 times and electron microscope magnifies the object over 2 lakh times. That is, magnifies says that means it is giving larger view of the object. It is making the object appear bigger or larger so that our eyes can view it. So the compound microscope magnifies the object to 2000 times bigger than the actual size of the specimen, whereas electron microscope gives 2 lakh times bigger or larger view of the specimen. So that is here you could see some cells and its structures, okay, whereas electron microscope can deeply give its structure, many other things present inside the specimen. So still more deeper details can be viewed using electron microscope compared to compound microscope, since there's difference in magnification. So these are the differences between compound microscope and electron microscope. And this compound microscope is used in laboratories, schools, colleges, and hospitals, etc. Whereas electron microscope is used in large scale laboratories, research centers by scientists and other things in order to do the research, because it gives inner details, and inner things can be studied under the electron microscope, uh, due to which, because it gives 2 lakh times the larger image of the larger view of the image object. So these are the main differences between compound microscope and electron microscope. I hope you have understood about this in this slide. This is the parts of the microscope. I will be explaining some main parts of it. These are the eyepiece. This is draw tube. This is the body of this body tube. This is the coarse adjustment. This is fine adjustment. This is the limb or the arm. Then this is the nose piece. This is objective. This is the stage. This is the slide. This rectangular thing is the slide. The circle thing is a specimen or the object which we are viewing. These are the clips to hold the slide properly. This is the condenser, this is mirror, this is foot, etc. So what is this eyepiece? Eyepiece is through which we see the specimen under the lens. That means our eye will be here. When we view through the eyes, we will first see here. So the eyes will be nearby. So that is why it is called as eyepiece. Okay, through which we see is, this is the place through which our eyes lies, or we see through this particular lens. So this eyepiece has eyepiece lens. Okay, and this is the body of it, and this is the objective. Objective has lens inside it, as we have eyepiece lens inside the eyepiece. Here also we have lens. Okay, the objective lens, why it is called as objective lens? Because it is near the object. See, we keep the object here, right? So it is near the object, that is why it is called as objective lens. This is near the eye, that is why it is called as eyepiece. Okay, then stage. This is the stage to keep the slide. Something must be there, right? So this is the stage to keep the slide and the clips to hold it properly, so that we should get a proper view, right? In order to make it still, not to move here and there, we use this clip. In this slide, this round color one or the circle one is the specimen, what we are observing under the lens. Okay, and next is the coarse adjustment. What is this coarse adjustment? This is a rotating knob, okay, where we use to move forward and backward. What happens if we move this knob? Here the lens keeps moving either towards the slide or away from the slide, in order to make the object more clear. We use this knob. Okay, the knob is used to move the objective lens. The objective lens here will be the lens, in order to move this near to the object, or away in the sense, away from the object is far from the object. Okay, either if we take sometimes far, it will be observed more clearly, or it is moved towards, so that we get a clear image, or I mean sorry, clear specimen view. Okay, so this is towards or away from the specimen being viewed. This fine adjustment is finally done with slight movement so that the object or the specimen is becomes clear. In order to move in order to observe this particular object or the specimen placed here should be clear, that means we use this fine adjustment knob in order to make it. This we move only slightly, whereas this coarse adjustment we move the objective lens little more so that to get the clear image. Okay, for example, you can take when taking the photo in the camera, you keep using that zoom, right, to get a clear photo. So for the same purpose we use this here as coarse adjustment and fine adjustment. Okay, then this is the mirror. What happens is, the in order to view the object or the specimen present inside the slide, we need light or brightness. Where will this brightness comes? When the light falls on the mirror, it reflects back so that the slide will become brighter. When the slide becomes brighter, the specimen will be more clear to be visible to our eyes. So that is why the mirror is used here. The mirror projects or reflects the light so the specimen is easier to see. Okay, these are main things which I have explained. Not much is there in your textbook. So now magnification. If the eyepiece lens, as I said, there will be one lens in the eyepiece also, and the lens will be there present on in the objective lens also, right? So this has some magnification like 10x, 20x, 40x, 100x, etc. Okay, that has a particular magnification. In the same way, even the objective lens has some magnification like 10x, 20x, 100x, 400x, etc. Okay, so based on the eyepiece magnification, we get here a bigger view. Okay, so say how to find out this. Say for example, if the eyepiece lens is 10x, if we are using 10x here, 10x lens, and the objective lens would be 40x, the total magnification we are getting will be, that means 10x into 40x, we get 400x. That is 400 times bigger it looks. Okay, that means the object appears 400 times larger than its actual size or the original size. The size may be very small, but using such magnifications, we get that much time bigger it looks, that much time larger it looks under the microscope. For example, the eyepiece may be 20x. Okay, and here it may be 10x. So how much? 20x into 10x gives you 200x. So that much magnification or that much times bigger the specimen looks. Okay, based on the eyepiece lens and also objective lens magnification, our uh, getting the object clear would be deciding that only we use here that particular magnification so that to make the specimen more clear. Okay, and we get through these, that means here the object appears 400 times larger. It may be very small, but it looks 400 times bigger when viewed under the microscope. That is why the microscope is used in order to view very minute objects, since they are not visible to our eyes. To make it visible to our eyes, we are using such type of lens inside this microscope, and we are able to see some tiny specimens, tiny organisms, or tiny objects. Okay, these are some of the parts of microscope. Next slide. The cells and its types. The cells when viewed under the microscope. When viewed under the microscope, you can see here is thin lining, right? Thin black lining kind of, right? So the outline can be uh, of membrane. Since it is in the cell, it is called as cell membrane. It's thin outline, right? It is called as membrane or the cell membrane here. Here also you can see lightly it is visible. Okay, so these thin like outline is known as membrane, that is cell membrane. And the inner pinkish color, here also you can see inner some dotted or jelly like thing, that is cytoplasm. And here the middle dark purple color circle, dark here also it is somewhat brown. Okay, dark something dark circle or dot like structure is known as nucleus. Okay, when viewed under the microscope you could see these basic structures or these basic parts. They are cell membrane, the cytoplasm and nucleus. Okay, the cells need not take the same shape or size of as I have shown here. It can take different shapes and sizes, as I have shown here, types of cells. There are trillions of cells present in our body and in other microorganisms, I mean, in other organisms. One such types are here, mentioned here are muscle cells. See, they take the shape of this kind, and the fat cell look this, and the bone cells take the shape of this, blood cells, nervous cells, epithelial cells, etc. These are different shapes and the cells have different shape and sizes. Okay, it need not have always this, as I've shown in the slide. It can take different shapes. So there are some of the types which I have shown here. Okay, then uh, when I see these many things present inside the cell, there are when I if I say this is viewed under compound microscope. Okay, when I view one particular cell under electron microscope, this would look like this. Okay, can you imagine now what is the difference between compound and also the electron microscope? So one such cell in the compound microscope may look like this only basic parts. Whereas when observed under electron microscope, it gives a still more detailed study, or it gives a still more larger view, right? When viewed under one such cell, looks these many things, right? So the cell not only contains the cytoplasm, nucleus, cell membrane, it also has so many things inside, right? When viewed under electron microscope, right? So many things are there inside, not only these things. Now cell has special components, as I said, membrane, cell membrane, cytoplasm inside, and also the nucleus. It also contains cell organelles. Okay, the cell organelles are, for example, mitochondria, endoplasmic reticulum, chloroplast, etc. All these are some of the cell organelles. What are these cell organelles? Let us study in the further sessions. Okay, so these cells has special components along with the cell organelles which perform specific functions or special functions. They perform functions. Each and every organelle do some its own work, which has made a cell a structure and functional unit of life. So all these together work and make the cells a functional unit of life. Okay, the cells contain special components and also the cell organelles which perform special functions that made or that has made a cell a structural and functional unit of life. Okay, let us study some more things in the next session. So thank you, children.